In the first position, I would go, first position is a U in the codon, so I'd go to this box. Second one is a U, I'd be in that one. And then second position's up here. Third position is indicated within the box on the right-hand side. When I have a U, U, C, U, U, I'm in this quadrant. C is the second position. That goes for the amino acid phenylalanine. So you can look this up for anything. Any of the 20 amino acids. Several of them are highlighted in color. These are special positions. Proteins always start with a methionine. That's the start codon, AUG. And there are 3 codons that serve as stop signs: UAA, UAG, and UGA. So when those come into position at the ribosome, if you have a UGAs show up at that spot in the ribosome, then special things happen to stop the protein synthesis machinery, and it just cuts it off right there. It's kind of interesting that all you've got is a stop and a start signal. There's no other punctuation. If you think of this as a language analogy, there are no commas, there's no parentheses, there's no spaces. If you think of these as words, you know, that this codes for methionine, that goes for proline, you kind of, if you're writing words on a page, you put spaces between the words, the chunks that have meaning. That doesn't happen here. It's just, this is the way it appears into the cell. I've highlighted it here with slashes to show the codon structure. But the cellular machinery has to kind of search and find, oh, that's AUG. So this is the register I must stay in. I'll read these in groups of 3. If it gets off, if it slides off course by 1, let's say it starts there, and then for the next one, instead of reading that tree, if it skipped one and started reading in that phase, everything would be mutated after that. So it's got to carefully stay in register. There's no marks or gaps or anything to help it stay in that way. But this particular guy, if you recognize that as the start codon AUG, you could mark it out that way, and you could see, using that table, that would code for that peptide, the methionine, proline, or valine aspartic acid. Okay, so in a regular biochemistry course, we'd spend probably the whole quarter on information transfer. All the stuff we're talking about today, way much beyond that. It's not really too appropriate for this course, but you guys do need to know about this stuff. There are kinds of information that overlap with the field of nutrition, which is why we didn't eliminate this topic entirely. I'm just going to highlight 3 quick areas here, just to point out some of the ways in which this kind of stuff might be of interest to you. We'll look at manipulating DNA, genetically modified organisms. We'll look at one genetic disease, area, and this topic of post-translational regulation. There are many ways to regulate gene expression, and I'll highlight just as one. MicroRNAs. It's kind of a hot field these days. So miRNAs means micro, not mRNA, which is messenger, but miRNA. These are tiny little chunks of RNA that we all make. They're non-coding, that is, they don't code for the production of a protein. They just are made, they're trimmed up a bit, and they exist to regulate other messenger RNAs. We make at least 1,000, probably more. Some people estimate that most of our genes are regulated by them. And different kinds of tissues, like liver versus brain and so on, express different ones. And it's involved all kinds of things. Cancer, various disease states, but normal regulation. So in the nucleus, these things are made as short segments. They might have maybe just as few as 20 bases in them. They're exported out into the cytoplasm. They're chopped up a little more so So these are very small segments of RNA, these microRNAs are. And they bind to proteins, in this case it's known as a RISC complex. They use Watson base pairing rules, Watson-Crick base pairing rules, to come up to messenger RNAs that are out of solution. So out here in the cytoplasm, you have to think that in a normal cell, proteins are being made all the time, ribosomes are busy, messenger RNAs are floating out here ready to be copied into protein. These microRNAs, which are associated proteins, will bind to some of those and kind of tie them up and prevent them from undergoing translation. So
I'm sorry, do they bind to the protein that's been made or to the
No.
mRNA?
to the mRNA, they're using Watson-Crick base pairing, that molecular recognition of A's to use and G's to C, to say, oh, I know this messenger RNA. And it's not just any messenger RNA. These are sequence specific, so there'll be a microRNA for one gene, a different microRNA for a different gene. So what will happen is it will bind to the messenger RNA and stop it from ever being used by the ribosome. So this is a way of slowing down production of certain proteins, depending on which of these are made and how many copies of them are in the cell. So as I say, this is a real hot area of research right now. It looks like, you may have heard of junk DNA, the stuff between genes. We have tons of it. There's way more junk DNA than coding DNA. This is some of the stuff that's in the junk DNAs, these microRNA genes. They don't code for big proteins, they code for these little tiny bits that seem to play a role in controlling how much of each protein gets made in a cell. Now, all eukaryotes have them, plants have them, animals have them. In plants, when these microRNA things bind, the associated RISC protein actually chops up the messenger RNA. So it actually hunts for it, finds the messenger RNA, and destroys it before it can be used. In animals, that can happen, but more likely, it doesn't bind as tightly, so it doesn't lead to digestion of the message. It just binds to it and keeps it inactive. So it kind of temporarily ties it up. But doesn't digest the target messenger RNA.